So we're starting the second round of oral presentations. These are the more scientifically focused ones. And our first speaker is Yue Zhang. He is a postdoctoral fellow in Ivan Pedroza's group. Ivan Pedroza is his mentor. And the title of his talk is Exploring Intratumoral Heterogeneity of Lipid Metabolism in Clear Cell Renal Cell Carcinoma with Dixon-based MRI.
Thank you for the introduction. Good afternoon, everyone. So, renal cell carcinoma is a disease composed by many tumor subtypes. Clear cell RCC is the most common subtype, representing about 75% of renal tumors. We now know that clear cell RCC is heterogeneous with multiple mutations, leading to different biological behaviors. Fatty acid synthase is a key enzyme in the biosynthesis pathway of fatty acids. This paper from the Cancer Genome Atlas group has revealed that the upregulation of the genes that control this enzyme correlates with worse prognosis. Therefore, we hypothesize that the lipid accumulation in clear cell RCC correlates with tumor aggressiveness. The development of a non-invasive imaging approach would be valuable for in vivo tumor characterization. Compared to tissue biopsy, imaging has the potential to overcome the limitation of sampling errors in heterogeneous tumors. Multi-echo Dixon-based MRI is a method for fat fraction quantification. On fat fraction map, the value of each voxel is the percentage of the signal out of the total MR signal that comes from fat. This technique has been validated in liver steatosis against tissue biopsies. Therefore, the purpose of our study was to correlate in vivo Dixon-based fat fraction measurements with tumor aggressiveness evaluated by Furman grade in clear cell RCCs. We also wanted to assess the intratumoral heterogeneity of lipid accumulation in clear cell RCCs. We also want to correlate Dixon-based fat fraction with lipid accumulation in tissues. This is the general workflow of our study. So the patient underwent MR imaging, including a M-Dixon acquisition for fat fraction quantification. A systematic ROI analysis was performed to measure the fat fraction for each tumor. The patient then underwent surgery to remove the tumor. Each tumor was classified based on Furman grade, and we studied the correlation between fat fraction and Furman grade. Second, from fat fraction map, the target ROIs were placed on representative locations within the tumor. Meanwhile, tissue samples from the same location in the tumor specimen were collected as target samples. These target samples underwent all-red-O staining or mass spectrometry, and the results were correlated with fat fraction. In order to assess the intratumoral heterogeneity of lipid accumulation, we perform a systematic ROI analysis by placing 8 ROIs within each tumor. 4 ROIs were placed in a superior slice of the fat fraction map, while the other 4 were placed on an inferior slice. The mean and standard deviation of 8 ROIs were calculated as representative values for each tumor. All-red-O staining allows histology visualization of intracellular lipids, which appears as red areas on histology slice, whereas blue dots indicates the nuclei. The percentage of cells stained with all-red-O was estimated visually to the nearest percentage by a pathologist who was unaware of fat fraction results. This is an example from the systematic ROI analysis. The values on the fat fraction map are the fat fraction for each ROI, and you can see how different the lipid accumulation is in different regions in the same tumor. This analysis was performed in all the tumors, and we found a heterogeneous lipid accumulation among different tumor grades. and also heterogeneous in each grade, and also even in each tumor, as indicated by the large standard deviation in many of these cases. However, we did not find a statistical correlation between fat fraction and the tumor grade. Here, each point represents the result from a target ROI on fat fraction map and its corresponding already percentage in fresh tissue samples. The Spearman correlation shows the in vivo fat fraction measurements significantly correlated with already staining at histopathology. Here, in this tumor with a lower fat fraction, the orange circle indicates the target ROI with a fat fraction 2%, and the already stained about 5% of the tumor cells. In another tumor, the fat fraction was high, about 14% in the orange ROI, and already stained about 90% of the tumor cells. So this is the intratumoral heterogeneity. We also observe a heterogeneous lipid accumulation in the same tumor, as in this case, 2 representative areas were selected based on fat fraction map. One was in a high-fat area with fat fraction 15%, the other a low-fat area with fat fraction 2%. Correspondingly, two fresh tissue samples from the same location showed staining in 70% of the tumor cells in high-fat area and 30% in low-fat area. From mass spectrometry, the amount of cholesterol and triglycerides in tumor samples was largely increased compared to their amount in normal kidney. The Spearman correlation showed that the in vivo fat fraction measurements significantly correlated with cholesterol and the triglycerides. Limitations in this study. So the misregistration between MR images and tissue samples can lead to inaccurate correlation. Therefore, we have implemented a protocol using tumor modes generated from 3D printing to facilitate the tumor orientation. Second, we are also working on assessing the fatty acid synthase expression level in our tumor samples. In conclusion, the in vivo Dixon-based fat fraction measurements did not correlate with tumor aggressiveness, as indicated by Furman grade in Clear Cell RCC in our patient. The lipid accumulation was heterogeneous among different tumor grades, and also within the same grade, and also among different regions of the same tumor. This might provide an opportunity to further understand the metabolic alterations in Clear Cell RCC and the potential correlation with clinical outcomes. We also validate our results by confirming that the in vivo Dixon-based fat fraction correlates with lipid accumulation indicated by already staining, and also find a correlation with lipidomic results from the mass spectrometry. Thank you.
Are there any questions? There's a question, Raythan. Wait for the microphone.
So I'm very curious why you did the heterogeneity analysis just selecting 2 axial slices or 2 levels rather than the entire tumor.
Because we have noticed that the lipid accumulation is very different even in the same tumor. We are trying to use a method that can select different regions in different tumor. So that's the approach we are going. So we place 8 RIs in 2 slices. One is, but those 2 slices are in different regions.
But don't you think you're missing other parts of the tumor?
Yes. Yeah. This approach has the limitations. We also thought about maybe later we can do maybe a 3D histogram for the entire tumor to evaluate that.
Yeah, I think that's probably the best method in PET. That's what we do the entire tumor analysis.
Thanks.
Neil?
Thanks. Great presentation. Furman grade is determined by a pathologist, a single pathologist, and I'm wondering if you can challenge the pathologist to do a variance read to see what is the distribution of their agreement. And then the other thing is Furman grade is it's a indirect readout of aggressiveness, and I'm wondering if there's an opportunity to look at further downstream outcomes because you may have more important correlations than could be realized with pathology.
Yes, first, for the first part, the pathologist was evaluated by a single pathologist in our study right now. We haven't evaluated by the agreement by two different pathologists yet. That's a very excellent point that Furman grade is not really a gold standard at all, but we are also working on trying to correlate our fat fraction measurements with the later clinical outcomes. But right now, our patient population only within maybe 2 years. After a while, we might have enough data to do that correlation. Another thing is we are trying to correlate with between the fat fraction and the genetic profile. That might provide additional information for that.
Good. Thank you again.
Thanks.